Good evening, everyone, and I would try not to hold you back from your weekend, relaxing weekend. And what I would like to do is just give you a thought to your imagination, because what I'm going to present today is something that you are going to see 5 years down the line. A decade back, a genetic disorder was rare. Genetic was something which was rarely seen and talked, observed, noted, prescribed, and everything. But today, a day of OPD without a genetic disorder is a rare thing. So that's the commonality that we have raised to, we have come to. From few to few hundreds to few thousands of genetic disorders have been now got into our daily practice, daily routine practice, from skin to from inflammation, from MD medicine to habitual insult to IVF oncology. So everyone in their family, maybe close kin and court, have at least one incidence of cancer or infertility or neurological disorders or an habitual insult or something like. And all these are connected to genetic disorder. Now, one thing about genetic disorders are, so far it is known to be a non-curable part. But soon I will be talking about something which is giving a way that it is also curable. Next, please. So this is the basic DNA, and everyone knows about it. It's the sequence which makes us what we are. This is the sequence which makes the life what it is. And this change in the sequence, next please. This change in the sequence brings about a clinical phenotype, and this clinical phenotype is known as a mutation, which is going to result into an adverse effect, which may result into hospitalization or which may result into some kind of treatment requirement. So this is a classical example of many genetic disorders. Today I'll be talking more about the hemoglobinopathies, because the topic connected, the CRISPR-Cas technology, is more applicable in current state to hemoglobinopathies like thalassemia, sickle cell disorder. You can see an heterozygous mutation above and then homozygous mutation. The lower one would result into a sickle cell disease. The upper one would be resulting into a carrier, which when marries to a carrier can produce at least 25% chances of having a sickle cell disorder. Next. So what is CRISPR? CRISPR is nothing but a clustered, regularly interspaced, short palindromic repeats, a repeat sequence. So what is CRISPR? CRISPR is a technology which has been very recently brought into healthcare, and it has given the ability to edit the gene very specifically and very accurately. So this ability to edit the gene in a very peculiar and a very specific manner gives a way of a complete new treatment methodology. Next, please. So what is like the discovery of CRISPR as the classical many good technologies which has come? It was first reported in 1987, but went into the carpet, went under the carpet, never came out. Till 2005, it was again reported, and till 2012, when Jennifer and Emmanuel reported it as an efficient technology for editing and got a Nobel Prize and opened up a new era of genetic editing. And this has entered health segment in recently 2020s. Next. So what are the important components of genetics? Next, please. Next. Yeah. So there are three major components. That is Cas9 protein, the guide RNA, and of course the PAM, or the prospector adjacent motif. So these three come together with a group of proteins, gives us that ability to do a molecular editing of a small part of a gene or a particular point of a gene in a nucleotide base pair and change the gene, which rectifies from abnormal to normal genomic sequences. Next, please. So this is how it works. So first is the guide RNA carries this entire complex to the right area, making it specifically bind to those areas where the editing is to be made. Once it finds the complementary sequences, binds to that area, the Cas9 and the CRISPR come together, they engulf the targeted DNA and then start the nicking part. Once the nicking is done, they change the base pair and edit it in a very specific manner, ensuring that the change is only in the targeted area and not anywhere else in the genome. We have billions of base pairs. We don't want anything else to be changed. Otherwise, we will end up in resulting into changing something else which we do not want to, and leaving behind what we wanted to change. Next, please. So this was the classical hypothesis which was put forward in NIH, that if Suzanne is affected from SCD, what we can do. So the neck is mold, the guide RNA carries to the particular position. Through the molecular mechanism, the enzymes cut the DNA, integrate the DNA with the changed base pair that we would require. And once that is done, next please. Once that is done, that autologous cells of the same Suzanne can be given back to her and making her more healthier than what she was before. When I say more healthier, making her less dependent to transfusion. Next. So I will be talking about SCD, that is sickle cell disease, and thalassemia, that is transfusion-dependent thalassemia, TDT. Next. We know the morbidity and the average lifespan is about 48 years, and there are millions of rupees which has to be spent in the caretaking of thalassemia when the child is having both mutation allele, that is thial major, and the transfusion dependent is from 2 to 5 weeks. So every alternate month we have to change the entire blood set of the the child, and that goes through a huge financial as well as physiological trauma to the family and to the child. So what can be done? Next, please. So what can be done is we can help them doing small experiment, what we envisage with our center, with the Indian Red Cross and Thalassemia Society of India, to give an opportunity to those children who are less than 8 in age, who are highly transfusion dependent, and we want to see whether this bone marrow can be taken, edited, and given back to the child, and thus ensuring that the child can be made less transfusion dependent from monthly to yearly ones. The target is yearly ones. We are not still sure about it, the outcome. We'll have to follow through the study for at least 5 years and see the promises that we are trying to make to that family can be ensured or not. Please, next. Next, please. So I'll go through how this can be done. There are two methods where gene addition and gene editing, both can be done. Gene addition, next please, through just addition of a normal gene, of a globin gene, ensuring the globin are produced in a right way. And next, the editing part, next please. The hemoglobin, where we have 2 alpha, 2 beta chains, and ensuring the fetal hemoglobin can be produced in higher sequences, higher concentrations, so that the altered hemoglobin, the beta globin chain, can be sufficed. Next, please. So we here in the gestational age, when the fetal hemoglobin goes down and the adult hemoglobin goes high, that is because of an expression of BCL11A that can be edited and changed, ensuring the continuous production of fetal hemoglobin and ensuring a good oxygen being transported from lungs to every part, every tissue of the body. Next, please. So this is the classical path, how it happens. Guide RNA carries the entire complex to the area where we are interested to nick and change the DNA. It binds to that area very specifically, ensuring that there is no other part of the genome being edited, and then finally changing the DNA from abnormal to the normal one, resulting into a normal globin gene, resulting into a normal kind of oxygen carrying capacity for that particular child or that particular individual. Next. So this is how it was worked, and Nobel Prize was awarded for this hypothesis and validated hypothesis. Next, please. So in our lab, we are trying to do this. We are not actually trying to change the globin. We are trying to change the expression of the BCL11A, and once the BCL11A expression is changed, that can ensure the continuous production of fetal hemoglobin. Next, please. So this is the flow chart that we are planning. Collect the blood or the bone marrow, edit it, ensure, freeze it, validate all that everything, the other part of genome is accurate and not have changed, and then again transfuse it back to the individual, ensuring that the severity of transfusion or the severity of frequency of transfusion can be reduced down. Next, please. So this was the first paper in 2016 which explained. Next, and next, please. One more. So I'll take you to the prime thing, or we'll take you to the crux of the entire talk. Next, please. So this is now US FDA approved. So US FDA first approved it for the SCD treatment. Now there are at least 12 such therapy lines which are under FDA approval, and sooner or later you will be seeing a lot of such US FDA approval genetic editing tools available in the field of therapy for at least known and established genetic disorders. Next, please. So in summary, CRISPR is a very powerful tool, of course, to be used in caution with all the ethical, moral, and clinical practice to be taken care of, and ensure that we try to give whatever best of the scientific knowledge that we have acquired in last many decades to be given to the family who are affected with genetic disorder. So this is my team and my collaborators. Thank you for the opportunity. I would love to thank the organizing committee, the Medigens team, for such a wonderful conference, for a wonderful conclave which has been made feasible by them. And thank you all for being attentive.